So this is spring 2020. We're working on microbial genetics. The last video we did was talking about transcription in bacteria, E. coli. And we said that transcription was the first step in a process called gene expression, where the information, encoded in DNA will be transcribed and then translated into a protein. So again, we're using E. coli as our model organism. So recall that transcription occurs in the cytoplasm of E. coli, of bacteria, and we'll also see that translation is going to occur in the cytoplasm of bacteria. So we're going to describe the process by which bacteria take the information encoded in the RNA sequence, base sequence of messenger RNA, and use that as a guide to make proteins. And this will occur at the ribosomes. So if I ask you where in cells does translation occur, translation is just a fancy term for protein synthesis, then you would say the ribosomes. And remember that bacteria have the smaller 70S cytoplasmic ribosomes, and eukaryotes will have the larger 80S cytoplasmic ribosome. So again, we're going to be focused on bacteria. So let's see if we can go full screen. So we had mentioned previously that translation is a really complex process. We're changing language from nucleic acid language to proteins. So we're going to tackle this as if it was an onion. So we're going to look at kind of like the simplest part of translation, protein synthesis, and then we'll start peeling back the layers of the onion to look at more and more detail. Okay, so translation, again, we're talking about how our mRNA will act as a template to guide protein synthesis. So how will the mRNA be translated? So it'll be translated, the mRNA will be translated as codons, and a codon is 3 nucleotides. So if we think of each of the bases as a letter, then these codons would be like the words, all right? And we'll use multiple examples here, you guys. So translation of mRNA, it begins at what's called the start codon, which is the first AUG that the ribosome encounters. And then translation will stop at 3 non-sensor stop codons. So we can see here UAA, UAG, UGA. These are all stop codons. And the key to translation is the genetic code. So let's take a look at this fantastic genetic code. So the genetic code is a one-to-one relationship between mRNA codons and amino acids. So on your lecture exam 2, I'll give you this genetic code table so you don't have to memorize which mRNA codons will be translated into which amino acids, but you have to know how to use the genetic code table. So you'll see here, folks, this table has all 64 3 base codons that we would find in mRNA. And remember, folks, in RNA we don't have thymine, we have uracil instead. So in the codon, each base has a assigned number. So this is base 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, right? So one way to use the table, if I give you an mRNA codon, is you could look for the first base here, so the first position here, and then you, so let's just say, for example, you guys, I gave you the codon CAA. So you'd say, okay, here's C, that's the first codon. So I know it's somewhere in this row, and then CAA, A is the second one, so I know my codon is going to be the junction. Here's the A in the second, here's the C in the first, and then you can look over here for the third base, or you can just use inspection. So here's CAA, and this is telling me the mRNA codon CAA would be translated. This is an abbreviation for a specific amino acid called glutamine. So on our lecture exam, I'll use a table that will have the full name of the amino acid. So I'd like you to use the full name of the amino acid. You don't have to memorize the abbreviations. Again, on your exam, we'll use a genetic code table that has the full names of the amino acids. So what this means is when a ribosome encounters the codon CAA on mRNA, the cell is going to translate this mRNA codon into the amino acid glutamine. Okay. Now, some specific, kind of important special codons. The first AUG the ribosome encounters is called the start codon. And in eukaryotes, the start codon AUG would be translated into methionine. In prokaryotes, it would be translated into a modified methionine called formal methionine or FMET. Now, any subsequent AUGs, the ribosome encounters, would simply be translated into methionine if it was a prokaryote and methionine if it's a eukaryote. The three stop codons we mentioned, UAA, UAG, and UGA, when the ribosome encounters a stop codon, the ribosome releases the mRNA, it releases the protein, and translation is stopped. It's halted. So these are sometimes called nonsense codons because they won't be translated into any amino acid. So again, the genetic code is a one-to-one relationship between codons and specific amino acids. So if there's 64 total codons, three of them are stop codons. That means 61 of them are what we call sense codons, and they'll be translated into 20 or 21 of the amino acids that we find in proteins. The key to this whole thing, the actual translator molecule we'll see later, is transfer RNA, and we're going to see transfer RNA can bind to complementary RNA. Okay, and we'll see this. tRNA carries what's called an anticodon that will bind to complementary RNA. And the tRNAs will carry a very specific amino acid. So they are the link, the bridge between RNA and proteins. We mentioned the three stop or nonsense codons. We mentioned the start codon. In bacteria, it's translated as formal methionine. In eukaryotes, it's translated as methionine. And the genetic code is described as being degenerate. And what we mean by degenerate is that if we take a look, let's look at the amino acid. This is leucine. LEU is leucine. You can see there's 1, 2, 3, 4, 5, 6 different mRNA codons that will be translated into the same amino acid leucine. And it's thought that this provides protection against, remember the mistakes that DNA polymerase makes? It doesn't make many mistakes, but it still makes mistakes. Well, it's thought that this degeneracy protects the cells from harmful effects of just those spontaneous mutation that occur when DNA is copied. And certainly if there's like mutagens present, we get increased mutation rates. The degeneracy would help prevent a totally different amino acid for being substituted, say if there was a mutation. So as an example here, you guys, if a cell, let's say the wild type, the original mRNA codon was UAA, but it experienced a mutation in which the mutant mRNA, the codon was UUG, you could see that there wouldn't be any change in the amino acid that would be chosen during translation. And again, folks, down here we can see there's actually 4 other mRNA codons that if these arose through mutation, they would still be translated into leucine. So the protein amino acid sequence wouldn't change, and that means the structural conformation wouldn't change and the function wouldn't change. So that would be an advantage. This, folks, is just to practice. This is the kind of question I'll ask you on lecture exam 2 in the short answer section. I'll give you a DNA sequence. We know this is DNA because it's got thymine here. So I'd ask you to transcribe this DNA sequence into an mRNA sequence. So base pairing rules, folks, between DNA and RNA. So thymine in DNA will base pair with adenine in RNA. Adenine and DNA will base pair with uracil in RNA. Cytosine in DNA base pairs with guanine. Again, here's the same thing. Cytosine in DNA base pairs with guanine in RNA. Adenine, this is the only tricky one, you guys. So adenine and DNA base pairs with uracil in RNA. This is the same. Thymine and DNA base pairs with adenine in RNA. And adenine and DNA base pairs with uracil in RNA, right? Okay. So this would be your transcription product. If I ask you to transcribe this DNA sequence, this is going to be your transcription product. We'll presume this is going to be mRNA. And then what you'll do, you'll have the genetic code table stapled to your exam. Then I'm going to ask you to translate your mRNA product here. So let's just try this. So let's take the first codon. Remember, this is position 1, 2, and 3. So position 1, where are we going to find that? Okay, here's the first position. We're looking for A. Okay, so we know our codon is going to be in this row. The second position is U, right? So here's U. So we find the junction between A and U. We know our mRNA codon is going to be here. And again, folks, we can just use inspection. So AUG, there's our third position right there. So AUG, if this is the first AUG encountered, and if this is a bacterium, it would be translated into formal methionine. Now, if you just put methionine on the exam, I'll give you credit. If I tell you this is E. coli and this is the first AUG, if you put formal methionine, you'll get maybe like half a bonus point. So now let's translate the next mRNA codon, GGU. So here's G, and then G, and then here's the second position G. So GGU is going to be translated into glycine. Okay, good. And then CAU, here's C, A. CAU would be translated into histidine. Okay, so you would write out your amino acids here, and then you'd be finished translation. Now, the key to translation, the real translator molecule in the cell is called tRNA or transfer RNA, but I think of the T as being translator molecule. So it's the actual translator molecule of the cell. So you can see that it's single-stranded DNA, but it has this, again, complex 3D shape. It's a functional conformation, and its 3D shape is stabilized by hydrogen bonds between complementary bases, and it forms these cool what are called stem and loop structures. Stem and loop structures. This is simplified. This is a little bit more complicated. In class, I'm going to even simplify this cartoon even more. I'm going to make it look almost like a fork. So I'm just going to have in my little drawing, my simplified drawing, I'm going to have this like the handle of the fork, and then down at the bottom there's going to be 3 little prongs, and the 3 prongs represents this region of the tRNA called the anticodon. So the anticodon will be 3 bases, and they're going to bind to complementary bases on mRNA. So the anticodon will bind to complementary codons on mRNA. So we have one end here, the anticodon, that will bind to complementary mRNA codons, and the opposite end here is going to carry a very specific amino acid, and we'll be able to predict which amino acid a transfer RNA will carry based on which codon the transfer RNA will bind to. So if we look at the mRNA codon that a specific tRNA binds to, then we'll know which amino acid it should be carrying. Now, there's another concept that's often referred to as wobble. So when we look at the 3 bases of the anticodon, 1, 2, and 3, binding between the first 2 bases and the mRNA will be really important. Sometimes binding by the 3rd base isn't that important. We'll see this is, how should we say that, like the significance of like a perfect match between the third base on the anticodon and the 3rd base on the codon of the mRNA maybe isn't as important as the binding of the first 2. Excuse me. So what's really important is that the correct tRNAs get loaded with the correct amino acids. That's going to be really important. And as we might guess, there are very special enzymes in the bacterial cytoplasm called aminoacyl-tRNA synthetases, and their job is to correctly load a tRNA with the correct amino acid. Okay, so enzymes are going to load the tRNAs with the correct amino acid. So folks, here's lots of practice. These are like practice lecture exam questions. So let's see if we can't figure this out, you guys. So if a tRNA anticodon is UAC, which amino acid will it carry? Well, the trick here is to ask yourself, the tRNA anticodon UAC will bind to which mRNA codon? So we just use complementary base pairing rules. So anticodon UAC is going to bind to mRNA codon AUG. So AUG, that tRNA would be carrying methionine. If the tRNA anticodon is GGA, Which amino acid will it carry? Okay, we find out which mRNA codon will it bind, and the mRNA codon it would bind to is CCU. So let's find CCU, CCU. Okay, so that tRNA would be carrying proline. In bacteria, which amino acid will the tRNA binding the first AUG codon carry? So that would be the F-met, okay, formyl methionine. If a codon is UUU, which tRNA anticodon will bind to it? So if this is the mRNA codon UUU, the tRNA anticodon would be AAA, okay, and which amino acid, oops, little typo there, would that tRNA carry? So we look for the mRNA codon UUU, and that tRNA would be carrying phenylalanine. And then which anticodons would bind codons UAA, UAG, and UGA? Well, remember, those are the stop codons. So there will not be a tRNA anticodon that would bind to the stop codons. That's going to stop translation, the termination of translation. The ribosome releases the mRNA and the protein, and translation is over. So we'll next talk about where translation protein synthesis will occur in the cell, and you know that that's going to be at the ribosomes. Okay, so Dr. Ken Naganuma described ribosomes at the cellular workbenches at which translation occurs, which is such a great description. So the ribosomes, folks, are made up of ribosomal RNA and proteins. Ribosomes have two subunits, a large and a small subunit. And the units we use to express the sizes of the ribosomes and their subunits is the Svedberg unit, and this is a unit that's used to measure sedimentation rates when we spin things really quickly in what are called ultracentrifuges. So small subunit, large subunit. So we want to remember that prokaryotic ribosomes are smaller, they're 70S, and eukaryotic cytoplasmic ribosomes are larger, 80S. And this is significant because we have antibiotics that at therapeutic levels will bind to inhibit 70S ribosomes such as bacterial ribosomes, but at therapeutic levels those antibiotics should not bind to our 80S cytoplasmic ribosomes. Right, so again, the 70S bacterial ribosome would be a unique target for our antibiotics. If we were to look at a ribosome in the process of translation, we'd see here, you guys, there's going to be like a channel, a groove that will bind the mRNA. And then if we were to cut the ribosome longitudinally, we'd see within the ribosome there are 3 binding sites, and you don't have to memorize their names, but I'll just give them to you. These are binding sites for tRNAs within the ribosome. So we have the A site, the P site, and the E site. And again, you don't have to memorize these names, but the A site is for the aminoacyl-tRNA binding site. The P is for the peptidyl tRNA binding site. The E is for the exit. This is where empty tRNAs are going to get kicked out of the ribosome. Okay. So for lecture exam 2, folks, I would like you to know the subunits for our 70S prokaryotic or bacterial ribosomes. The entire ribosome is called 70S. The small subunit is 30S. The large subunit is 50S. And I know, folks, a lot of times people think this is a mistake. But it turns out if you centrifuge the ribosome subunits separately, these are the correct sedimentation rates. But if you put the 2 subunits together and you spin the whole ribosome in the centrifuge, the sedimentation rate is 70S. So this is just, unfortunately, the sedimentation rates for the subunits, it's not arithmetical, I guess. They don't add up. So this is correct. This is not a mistake. And folks, this is some detail that if, and I probably will ask it on lecture exam 2, but they would be bonus questions, okay? So one of the bonus questions would be, within the bacterial ribosome, what part of the bacterial ribosome recognizes a special mRNA sequence called the ribosomal binding site? That's an mRNA sequence to which the ribosome binds, and then it starts scanning the mRNA, and as soon as it hits the first AUG, that's where the ribosome will start translation. So this ribosomal binding site, it's really important. We want the ribosomes to start at the beginning of the mRNA sequence for a protein. We don't want it to land in the middle of the mRNA and just translate part of the mRNA. So the ribosomal binding site on the mRNA, it's really important the ribosome can recognize it. So in prokaryotes, it's one of the ribosomal RNAs. So in bacteria, it's the 16S ribosomal RNA that binds to the RBS's ribosomal binding site. So there's our 16S ribosomal RNA, and it's in the small subunit. And then this, again, to me, you guys, is so cool. So then if we want to know what part of the ribosome forms the peptide bonds between amino acids during the process of translation, it's so cool. It's not a protein. It's a ribosomal RNA. So this is a catalytic RNA. So it's called a ribozyme, right? And specifically, it's the 23S ribosomal RNA in the large subunit that is responsible for catalyzing formation of those peptide bonds during protein synthesis. So it's ribosomal RNA. It's acting as a ribozyme. Oh, and here, folks, again, I guess I jumped the gun earlier. This is just showing the binding sites within the ribosome. This is the binding site for the mRNA, and then we have the 3 binding sites for the tRNAs. So as always, you guys, we're always really interested in these unique targets for our magic bullets, our antibiotics that will kill or inhibit the bacteria without harming your human patient. So the bacterial 70S ribosome is a great target for magic bullets. So when these antibiotics bind to the bacterial ribosome, it'll stop protein synthesis, and that will at least inhibit the bacteria, and in sometimes can outright kill them. And another thing, you guys, that's really fascinating with this, later on we'll be describing toxins that bacteria make, and we'll be talking about protein exotoxins that bacteria make. So at one of the infectious disease conferences, they were stating if you have a patient that's infected with a bacterium that's making these protein toxins, make sure to give the patient a 70S ribosome inhibitor as one of the antibiotics, because that will stop the protein toxin production. So even before you kill the bacterium, you can stop them from making their protein toxins. And I just thought that was just such a cool concept. So examples of 70S ribosome inhibitors, the tetracyclines, macrolides like erythromycin and azithromycin, chloramphenicol. Chloramphenicol they usually don't give much anymore because in some people it causes aplastic anemia, so that's, I'm guessing, kind of a last-ditch effort. And then aminoglycosides, folks. Aminoglycosides are pretty powerful antibiotics. Do have to be careful because they can be nephrotoxic, means they can cause damage to kidneys, so you'd be really careful giving them to patients with kidney disease. And also they can be ototoxic. They can cause damage to the auditory nerve. And I've read that's supposed to be reversible, but again, we just want to be careful with the aminoglycosides. Aminoglycosides, it's a big family. It includes antibiotics such as kanamycin, gentamicin. There's a lot of aminoglycosides. And not to panic, folks, you don't have to memorize the exact mechanism by which these 70S ribosome inhibitors inhibit the ribosome, but to me it's, this is always really fascinating. So, here's one, streptomycin. This can be used in treating, I believe this is going to be, can be used in treating tuberculosis. So an example of an aminoglycoside, the tetracyclines, and here's the chloramphenicol that we want to be really careful with. So folks, why doesn't, say, tetracycline or any of those other 70S ribosome inhibitors, why don't they kill us? So again, it's because of the difference between the prokaryotic and the eukaryotic cytoplasmic ribosomes. So here's our prokaryotic, our bacterial ribosome. In humans, you guys, in eukaryotes, this is the structure of our 80S cytoplasmic ribosomes. So again, the sedimentation rates don't add up, but our small subunit is 40S, our large subunit is 60S, and when you put them together and you centrifuge them, you end up with an 80S ribosome. So again, at therapeutic safe levels, those 70S ribosome inhibitors will only stop protein synthesis on the 70S ribosomes without harming our 80S ribosomes. So folks, what I'll do here is stop this video, and then the translation part 2, we'll just go through these cartoons showing step-by-step translation. Okay, but we'll stop here because that was really, we've cut right in that video, you guys, we covered the most important information. Okay, so, but we will do a translation part 2.